příště se můžete těšit na přednášku profesora Freida o nepravém příběhu Viničné a zamrzlá vonce po 20 letech. A teď už přijde do angličtiny a představím vám dnešního přednášejícího. So it is my honor to introduce you Professor Schulze-Makuch, who is currently at the University of Berlin, previously at Washington State University. He studied the geosciences and then moved primarily to the field of astrobiology. He's the author of nine books, mostly popular science books and science textbooks, and also one science fiction novel, and of course, great many scientific articles. And the most recent one is related to the topic of today, which is life in the driest places on Earth. It was published in PNAS 3 days ago, and it's related to the Atacama Desert, which you can see in the video here. So I'd like to thank Holder very much for coming here today. And now we can listen to this talk on testing the dry limit of life, and he will take us from the Pitch Lake in Trinidad through the Atacama Desert in Chile to the prospects of detecting life on Mars. So thank you very much.
Thank you. So we thought to put up the video that you get some kind of an idea how it looks in the Atacama Desert, but this the zoom in actually comes later, but you will get an idea. So if I understand this now correct, then my talk should come up. Yes, okay, this works. Make this from the beginning. Okay, still there. Okay, good. I thought I'd give you some kind of an overview about different places and about planetary habitability. And we will start with Earth at two very different sites. One in Trinidad, actually, and there's a desert too. And then we go to the Atacama Desert over here. This is our truck there, looking and investigating. And then we go to Mars. And we see how this is relevant to Mars. So we go first from a small scale, from 10 to 100 microns, because this is also the scale where the bacteria do this thing. Then we go to a rock scale, and then eventually we go to a planetary scale, to Mars. So how I would like to start is with an asphalt lake in Trinidad. And you know, that doesn't look like a desert if you start with it looking, because there's some water on it. But we are more interested actually in the liquid asphalt. And I can introduce to you, this is the liquid asphalt lake. You see there how big it is. And over there in the lower area over here, there is a liquid asphalt. And the liquid asphalt has actually a water activity of 0.5. For those that are not familiar, water, if it's 100% available to organisms, it's 1.0. So if you open the faucet, water comes out as 1.0. Salt water has about a water activity of 0.99 because there's a few molecules that are bound to the salt that, you know, living things can get. So 0.5 is very low. That is usually assumed that microbes need to have at least 0.7. So this is Pitch Lake. It bubbles there out. And it's one of 3 natural asphalt lakes. And we like to work over there because the 2 other ones are not that great. There's one in Venezuela still, but that is covered by 0.5 meter of water with crocodiles inside, malaria-infested mosquitoes, and drug dealers. So we don't like to go there. The 3rd one is on the Iran-Iraq border. Not the best place either. So we like the one in Trinidad. And you see one of our guides there puts that up. This is the stuff you're working with. And it is actually pretty nasty to work. This was our first team actually working there at the asphalt lake. Because if you try to sample there, you cannot sample like a water sample. You know, naively at the beginning we had this kind of water bottles. That doesn't work. Yeah? So basically how you sample, you take a latex glove and just grab into it and get it out, you know, and then you wrap it around or you use some other kind of a tool. So we start off taking samples at different depths. And like I said, the measurement actually was a water activity 0.5, so you wouldn't expect any life. But we like to go, or I like to go, where people said there is no life, to find life, you know. And so we started the investigation, and I'll walk you a little bit to how that successfully evolved as well. So the composition were asphaltenes, well, heavy hydrocarbons really, right? And with a lot of toxic metals inside, there were gas bubbles that were mostly methane, but also carbon dioxide and a couple of other hydrocarbon gases. So, so the next thing we measured was the temperature, 32 to 56 degrees C. So that was significantly warmer than the surrounding, but you know, not extremely hot. Water activity, our measurements were between 4.49, 0.65. Of course, that's over a square centimeter or something like this averaged. So again, you wouldn't expect any life with this water activity. However, what we found, you know, one of our colleagues had the amino acid analyzer, and we put that over there, and well, we find amino acids. So if there's no life, why are there amino acids? So we looked then further and thought, let's do some isotope chemistry and look at the fractionation rates, and they are negative. And that could indicate possibly microbial fractionation, but that was not conclusive. So we got different kind of interpretations. Nitrogen, sulfur were fractionated too, and so it doesn't tell us much. Let's go a little bit further. So what we did then was phospholipid fatty acid analysis. So the phospholipid fatty acids are those in the membranes of organisms. And with those we actually can conclude back on the biomass. If you're not a chemist, don't worry on those ones below that are only biomarkers. We're just looking at the biomass at this time. And you see the biomass is about 10 to the 6 cells per gram. If I go out here and take a sample, I'm pretty sure I'm about 10 to the 9 cells per gram, so 3 orders of magnitude higher. So that is, you know, an environment there's not many organisms in compared to our temperate environment, but nevertheless significant, right? So somehow they make a living there. We went a step further and said, okay, if there's life, what is exactly in there? So we did some DNA sequencing, and we found microbial communities are a whole different communities. The important thing here is that most of them were actually not methanogenic archaea. Archaea are kind of known to live in this kind of extreme environment when it's hot or toxic, and so they are very common. Most of them were actually not. Most of them were bacteria and used nitrogen or sulfur. And we found more than 600 new species. So that was a specific new environment. And when we looked a little bit closer at our sampling points, we could distinguish between archaea over here and bacteria over here. And you see that the bacteria are about 100 to 1,000 times more common than the archaea. Also, as explanation, OP is an oil well, and MV is a mud volcano. So that is already, you know, sometimes some people probably think you're crazy because I actually took a sample from an oil well, you know, and looked for microbes. But we did find microbes inside, right? And the mud volcano as well. And if we now look here at those plots of the archaea and the bacteria up in the archaea, these dots basically show from the genetic markers how related they are. And the PL is all in the pitch lake. And you see that some of them are more related to the mud volcano or to the oil well than to each other. That means wherever we look, and the sampling points were not very far away, we have a very distinct microbial community. So one has really nothing to do with the other, or not much to do with each other. So there are ecosystems that seem to be separated. And the archaea gave basically the same picture. So, so what we found out there, that there's island-like population that are apparently adapted to the individual environmental niches. Very close. Some of them were better related to some other locality of a tar lake or so than actually in the asphalt lake. So, but our biggest question was now, okay, we have a water activity of 0.5. How do those organisms make a living there? They can't get enough water. It is really a desert, an extreme desert. So would they use the hydrocarbons as a solvent for life? That was one hypothesis. And in a way, the most exciting one, right? Because we all depend on water, and all the organisms we know depend on water as a solvent. So if they could use hydrocarbons as a solvent, that would be very exciting. Or do they extract water from the hydrocarbons? There were, before our investigation, there were a few papers that showed that some organisms can do it. Even E. coli can do it. So that would be another way to get the water extracting in from the hydrocarbons. Or do they thrive in niche environments within the hydrocarbons that have a higher water activity? So that were the three competing hypotheses. So we try to resolve that. So what is the first step? Okay, we're looking at our organisms, right? That's how the surface looks. And what we found, that there were small little water bubbles inside the asphalt lake. Yeah. So this is how it looks under the microscope. And that's how the water bubbles looks. And what do we find there? We find microbes inside the water bubble. So the water bubbles were the smallest really ecosystem, and I think still known to date. And we published that in 2014 in Science. This is, you know, we were really excited about it. So you see a couple of pictures still, you know. There's a bacteria on the left that are just dividing, and some other bacteria in the water droplets. Again, some more bacteria. So you see, we were very excited. So the thing was now that we looked also what kind of microbial community was there. And we see that in the water droplets, there was quite a bit of variety, quite a lot of different species. So showing us that this is really an ecosystem. Again, we don't have to go, if you're interested, I can send you the paper of the different organism or species, since a lot of you are biologists. But we keep here now to the main points. And then we looked into the what are the metabolites. Yeah? So we used an advanced method with one of my colleagues. And you see now that most of the metabolites, the breakdown products, were in the water droplets if you compare to the oil or just the water extract. So that's where by far most of the organisms were in. Well, to convince the reviewers, we had to make a point, well, maybe that's just rainwater, right? Well, that is not the case. When we sampled the water droplets, and they're only a few microns big, yeah, a few hundred microns big, we made a chemical analysis, and they are salty. So they are very close to the seawater around there. So that means that they actually come from deep down. And they also have the same isotopic composition, meaning the inclusions there pointing to that is not rainwater, that is deep water that comes up. So we came to a model how the whole thing works. So we have over here our water saturation. The water comes up and getting mixed into the oil. And there's not much on chemical background known, But the thing is, that was an early representation how the geology looks there. And what I think how it looks like this is there's a big oil reservoir, and it pushes the oil up. It mixes with the water inside, and if this water goes through the oil reservoir, you get the pitch lake, or it goes into the ocean. If it bypasses the oil reservoir, then you have your mud volcanoes. The mud volcanoes, actually, we are still investigating, and we actually find foraminifera and some shells and so in the mud volcanoes. Actually, that would have been a good video too. About 2 weeks ago, one of the mud volcanoes erupted. So we're still actively working on the mud volcanoes. So we have those water droplets. Of course, they're living there in water, but it is a desert environment. And we also have some work still going on that some of the organisms may actually live in the oil. And that is, of course, very interesting. How do they make a living there? And it seems to be it has to do with the pirate clusters. We found that very cool when we looked through the microscope. It's like a spaceship moving through, right? And some of the sulfobacteria seem to be associated with those pirate clusters. But this is still ongoing as well, so we don't know that exactly, what is going on. So why is that important? You know, because of Saturn's moon Titan. Titan is a moon around Saturn, which has a thick atmosphere, actually, of 1.5 bars, and has liquid ethane and methane lakes on its surface. And this is a radar image of, actually, of Titan. And you see the ethane methane lakes. There's our pitch lake. And these are two lakes compared. This is Legea, and this is Lake Superior in Canada. Sorry. And you see how they compare in size, actually. You know, so these lakes on Titan are actually pretty big. So the Titan environment is how you can imagine, you know, in Antarctica or nowadays outside, if you have an oil spill. You know, you still not even get close there. But let me tell you, you have an atmosphere of about 1.5 bars. Most is nitrogen atmosphere, about 5% methane. And then you have all different kinds of organic hydrocarbons still in the atmosphere. You have methane clouds where it rains methane out of it in monsoon-like downpours. You also have acetylene that is forming in the atmosphere and trickling down and probably accumulating on the bottom of the hydrocarbon lakes because it's heavier than the ethane and methane. You also have geysers. You have probably underneath still an ammonia water ocean. Very cool things going on. But the most interesting where I want to point now is on the bottom of those lakes, because you have here the acetylene, and you could in principle have metabolic reaction, having that reacting with hydrogen to form methane. And remember, you have 5% methane in the atmosphere, so some of the methane could be forming biologically. If we hone here in, we see that it's even getting more interesting, because we have the multiple interface environments. We have the ammonia water ice, and if that is getting a bit heated, then it becomes a liquid, right? And that interacts with the hydrocarbons. Oh, thank you. Well, talking about the desert, yeah, right. So you have that actually is a mixture of water or ammonia water, which is even better. This gives you the nitrogen groups, right, with the hydrocarbons. And if you then still have some clays and some entrapments, then you have everything what we think was there for the origin of life. So this is actually one of my favorite places to go, Titan, because there you can see how far chemistry could evolve toward life. And this was what the place what I would suggest to investigate and figure it out. Okay. Unfortunately, I'm water kind of organism and not on hydrocarbons, so I need water. And over here, they need water too. So when I got the European Research Council Advanced Grant, it had a topic on habitability of Martian environments. Because we have to recognize Mars as a planet, right? It's not one specific environment. There's a whole different types of environment. And we see that Mars changed too. On the left side, you see Mars 4 billion years ago. Mars today looks much less friendly, but nevertheless, if I look at the Atacama Desert here in Chile and Mars Desert, it looks very similar. And when we were there the last time and took some pictures, I said, well, if one of us has an astronaut suits on, we can sell it we were on Mars. It looks that similar. And those boulders that you see, so they don't move by fluvial things, you know. Wherever we look here, we think, oh, it's fluvial. They move by earthquakes, because you have still relatively often earthquakes, and with that they're moving. So, but let's look at a little bit from a planetary viewpoint. Earth is a water world compared to Mars, right? So when we look at the fossil record, we see mostly ocean sediments, and there we find the fossils. If we look at land environments like soils, permafrost, ice, snow, regolith, usually the fossils are not preserved. So what we think is actually that the land was much faster colonized than most people think. It's just, it doesn't fossilize very well. And we know that early on we had a cold and wet Mars. You know, there was quite a bit of water on it. And that this kind of scenario applies, it's becoming more and more likely and has been proposed by several planetary scientists. So if that is the case, actually an early Mars biosphere would have enough time to colonize the land areas, right? And that seems to be the case because we have actually aquatic habitats on Mars during the late Noachian, that's about 4 billion years ago. We also have inferred existence of aquatic habitats about 3 billion years ago. Just the Curiosity rover found a lake that was about 3.6 billion years old. And episodic aquatic environments even during the Amazonian, so this is the latest period of Mars. So if we want now to find the dry limits of life, we have to see how this is affecting habitability. What is affecting habitability? And we have to go to those places where they're comparable. So in my view, there's actually only two really places that are comparable to Mars. This is the Atacama Desert, the hyper-arid core, and that is the dry valleys of Antarctica. And you will see the differences now. In Atacama, we have a mean annual temperature of plus 19 degrees C. This is fluctuating very much. The temperature is in the dry valleys, the mean annual temperature minus 20. So that is more closer to Mars. But on Mars, we actually can have temperature up to plus 25 degrees C too. But the advantage of the Atacama for most is that you have the same mineral deposits and geochemistry like perchlorates and nitrates that you find on Mars. The temperature fluctuations are in both cases pretty high. The mean annual relative humidity actually is smaller in the Atacama than in the dry valleys. And mean annual precipitation, you see too, is lower in general in the Atacama Desert. Smaller than 2 millimeters. So first of all, we have to understand the driest deserts on Earth to apply that then to Mars. So on Earth, the land dwellers would have to adapt to desiccation, high salinity, and UV irradiation. And we want to know what would be that limit of that adaptation and what is that limit on Earth. I mean on Earth and the general one, you know, how much life forms can get. So you start really out with arid. That is between 25 and 200 millimeter rainfall. So we see that some of the soils and habitats are colonized by cyanobacteria, especially in the top few centimeters. You see something like that, because you see the biological soil crust, and that can cover up to 20% of the surface. And you have still really fast growth rates, and you have colonies composed of lichens or mosses, mostly lichen. If you go drier, then the colonization becomes quite a lot more patchier. And you see a gradual decline of cell abundance and species diversity. And you see something like that, the hyperlithic community. I turned that quartz rock around. So this was on the bottom, the green. So they are living actually on the bottom of the rock. Some light is still going through here, so they can do photosynthesis. And that quartz rock nicely blocks out the UV radiation. And of course, on the bottom of the rocks, they have more moisture. So that is a microhabitat, so they can make a living there. And we studied those as well. If you go even further into the aridity, you achieve 1 millimeter of annual precipitation or less. Okay? And then hyperlithic communities don't work out anymore. It's still too dry for that. And what you have then is that the microbial respiration is undetectable. The microbial biomass has reached the lowest level that you can measure on Earth. And it is very tough for the organisms. And you see those are this kind of environments that look like that. However, even in those environments, if you know, as a geologist, you like, or with some training in geology, you like to do that, hammer off the rocks, yeah, the salt, you see something like this. This is like a greenish, right? So if that is not a fluoride mineral, it's photosynthesized, it's cyanobacteria. And they are cyanobacteria. And there's cytonomine, that is a pigment. So there's actually still life. So how do they make a living at those, when there's basically no rain? Well, those microbes have a special trick, because they use the hygroscopicity of the salt to attract water directly out of the atmosphere. So, so you have, by a process that is called deliquescence. So they are so hygroscopic that they dissolve into the water they absorb. So actually you get your little solution that way, right? And so we showed that some of them use a halite in the Atacama desert. That's the same stuff that you put in the morning on your egg, yeah? And when you think about it, when you see your salt dispenser, it has some rice grains inside, right? And the rice grains are there to suck up the moisture, because if you don't, you clump the salt. Try it out. So salt attracts water directly out of the atmosphere. And those guys use it. Even that guy uses it, the thorny devil, right? Some kind of a lizard. And he optimized it, but that doesn't live in the Atacama desert anymore. I just put it in as a principle way how to do it. So life is in that way smart. They find the ways. So what we found out is that there's a predictable sequence of ecological transitions that occur on land as conditions become increasingly dry, right? So you start out with your aquatic ecosystems, then you get your rapid growth, continuous colonization, what we showed under arid conditions. Then you become even more aridity, and you have your endoliths, your hypoliths, so the cyanobacteria that's inside the quartz or underneath it. And then you have your hygroscopic salts. Okay? So that is applying to Mars as well, because if you see there, the time scale, that's what we reached on Mars today. So if we compare with Mars today, and in the past we see that. In the Atacama, we have on average 1,200 precipitable micrometers. And the Earth's south pole is actually still a little bit less. So that is still quite a bit higher what we have today on Mars, planet-wide, smaller 3 to 50. However, Mars had, in the past, it had higher obliquity. And it was basically, Mars has no moon, right? Or just two rocks that are circling around that don't count as a moon. We have a real moon. Our moon is stabilizing our axis. Mars doesn't have that, so sometimes it just falls over, basically, right? So things change. The pole area becomes the equator and vice versa. So the water getting redistributed. If that happens, or through more volcanic exhalation, you actually get more precipitable microns, moisture, up to 3,000. Then you are there with the Atacama. You even have a little bit more. On average, it's still 2 to 50 times drier than the driest region on Earth. But nevertheless, we have to keep this in mind. So one of the things we came up then in our paper, in one of our papers was, well, if you look for life on Mars, think about land dwellers, not oceans, and don't follow necessarily the water, follow the salt, right? Because that is the last habitats on Earth where it works really well. And if we do that, where we should explore on Mars is the southern highlands on Mars, and not the boring equatorial regions where every lander goes down because it's easy to land, and the engineers tell us we can only land there. We are scared of the boulders, we are scared of the mountains, we are scared of lava tube caves. So we have to go there where it's interesting, right? So, so if we put our conclusions together on that part, we say that the habitability rules of land environments are fundamentally different from aquatic environments. Critically, land dwellers rely on atmospheric sources of water. And terrestrial deserts are a vivid example of that. And there's a predictable sequence of ecological transitions and that happen on Earth with increasing dryness and apply to Mars. So life on Mars need not to become extinct with the disappearance of water. And the habitability window of Mars may have closed much later than most people think, or maybe it's still there. So what we did then next is let's go and investigate that. And so we looked into places like this. And you don't see much life there, right? You shouldn't see any life, actually. That would be rather disconcerting if you find an antelope or anything like this. Because there isn't anything, you know? And if you go there, it feels very differently, you know? Over here, if you go outside, you see humans everywhere. You see plants everywhere. You see animals everywhere. So if you're over there, you don't see any animals. You don't see any plants. You only maybe see your fellow humans. But there's not many of those either. So it feels very different, and the only thing what you feel is really the wind. Otherwise, there's only rocks and mountains. So it feels strange. It feels very different, unusual. So we did then investigate it and put a huge research group basically together and to look at those places and see what we can find out. And our approach was a shotgun approach, basically. I guess I got that from being long time in the US. I thought, okay, let's take all the resources we have and throw everything at it and see what we find out. Okay? And the hypothesis was there, before we started that, is that the microorganisms that were detected once in a while, or DNA, was basically only from wind blowing in. They were falling down, and they're slowly dying there, yeah, like the early miners there. We found that. Actually, we looked at some firewood for a campfire and found that. We didn't touch it, but it was kind of tempting because we needed something to make a fire. And there's no wood anywhere, right? So otherwise. So we looked, and what we did specifically, we went to the driest places that we could find. And that is Yungay. If you look at this, at the landscape, I think it looks really like marsh. I mean, I can't see a difference, right? You have your boulder, and we looked in microhabitats. So we dug a hole. There are some of the gypsum nodules here coming through, and see whether we find something special. So, so when we did all the analysis together, the result was actually a paper that just came out 2 days ago. And if you look to the science news, this was quite a bit on the science news. And where we were showing that, well, no, they're not just only dying there. There's actually a habitat over there. There's a transient habitat. Because after a rainfall event, we had a rare rainfall event. And first when we went there, we said, oh shit, it's raining. Yeah. We're going to the Atacama Desert, preparing for months, want to go to the driest spot on Earth. And then 3 weeks before it's raining there, yeah? Once in a century. It's raining a little bit more often then, but this kind of rain event, we had the data from 1978 on. There's no data before that, you know? And that was the strongest rain event of all, you know? And so, okay. So we thought that was really bad luck, yeah? But in a way, it was a good thing, because it showed us then that, you know, there is still organisms that live there and do their things. And so we looked at different kind of indicators of activity too. So this is on the upper left is ATP, for example. The numbers is CS, this is coastal soil, from the more wet area to the driest area. And you see it's going down. This is what we expect, yeah? The phospholipid fatty acids too, the numbers is going down. But we do still have them. We still find them in the driest core. And the biomass is going down too, although there are some peaks corresponding to microhabitats. So we did several different things. We did enzyme activity. We did intracellular and extracellular DNA, intracellular, extracellular ATP, metabolites, and so I don't want to show you everything because if you're interested, just get the paper. I just want to show you still 2 things of the paper. That we actually used what is called genome-resolved metagenomics. So we were able, in the 2015 sampling period, to get some of the metagenomics out of it. This is more difficult because you need a little bit more for metagenomics than for the other methods. And then we could look into so deep that you were actually found some of the organisms are replicating. So with that we could make a good argument. They are living there. They use that area as a habitat. Not only have we several indicators of activity, but we also have of replication. And then what we did too still is compared from 2015 to 2017 with the following years we were there too. And you see in the upper left the water content is dropping drastically, what we expect. The anhydride content is going up. Some of the gypsum actually is becoming anhydride. So it's dewatering, yeah? So it gets out. And we see that the internal intracellular DNA, which is representative for the DNA inside the cells, or not outside the cells where the cell is dead, is actually going very much down. The intracellular ATP as well, but not here in the microenvironment. So with this is now highly relevant to Mars as well. And the reason is because even on Mars we have moisture event. We don't have a strong rainfall there. But we also have fog on Mars. We have near surface groundwater, so water on the mineral surfaces, and also water through what's called the recurrent slope lineae, what are probably briny salt water flows on the surface. And we even have an occasional nightly snowfall there, the microbursts. So we do have water events there. So if our microbes in the Atacama can survive there, then there's a good chance that the microbes on Mars can survive also. Of course, it's a little bit more hard, you know. Our Atacama guys, they are tough guys. They have a hard environment, no question. And now they have to give a little bit more UV on it, a little bit more lack of water, a little bit more chemical stress. And then perhaps they make it still or not. Of course, we don't know. But considering that Mars was wet and had oceans early on, I wouldn't underestimate the innovativity of evolution. And perhaps our Martian guys even are better adapted to the dryness and desertness than our Earth guys. Because on Earth, like most of us, live where there's a lot of water, right? There's only few spots where it's really tough, and the Atacama is the toughest. But Mars is a desert planet. So you would think if there was at some point a biosphere early on, that one would have much more evolutionary pressure to adapt to the dry environment. Here only one more image that shows that there is water on Mars. That is a gully as appeared through the orbiter in 2001 and that in 2005. So there must be, or the interpretation is really that there was water bursting out of the canyon wall and sublimated, and that's the salt left. So the water is there. So, and at the end, I just want to still run by you kind of a suggestion we had for a mission. And we call it locally target ecosynthesis, LOTES mission. And the idea was to engineer local habitable hotspots on the planetary surface to reveal any subdued biosphere that may be there. Okay, so in other words, we're speaking, if there's Martian microbes around, we want to make it nice for them, that they come out there and reproduce and do their things. And in order to do that, we have to make certain assumptions, you know. We need a liquid solvent. Well, since Mars is a terrestrial planet, that would be water. But, you know, on Titan, it could be something else. We need energy sources, we need nutrients. And there are limits exposed by UV ionizing radiation. And for Mars, that means lack of liquid water and high UV and ionizing radiation. For Titan, we did that too. That means something different. But we stay now on Mars. And of course, there's low amounts of organic compounds. So the absence, no one believes that anymore, but there's low amounts. So we call that mission DOMES, detection of Mars extant life in the subsurface. So DOMES would engineer the conditions for life in the hotspots, creating by placing small domes on the surface where liquid water and organic compounds would be supplemented. Okay? So the objective would be to generate confined warm and wetter conditions, but don't exaggerate it, you know, because maybe Viking didn't work that well because we actually drowned those guys, you know. If they are microbes that use the liquescence or water from the atmosphere, you know, and then you pour water on it, you kill them. And maybe that's what we saw. And you have to provide shelter against UV and ionizing radiation. So the requirements are actually not that bad, because you would have material used to shield the interior of the dome from radiation, but you have to let the visible light through. You must have a thermal insulation, or that would be good, so that the thermal fluctuations are not that extreme. You need a thorough sterilization before, because you don't want to get our Earth friends over there and grow them on Mars. You want to actually have the Martian organisms, if they exist. Implementation would be relatively easy. You just need to excavate with the rover. You can do just a couple of centimeters deep with scratching. And you could incorporate it in any of the rover missions also, and not much is needed. And to give you now an idea how something like this works, I show you that. I'm saying, what is this? Well, that is in the Namib Desert. And the principle is the same. This is a sandwich bag from some guy who just tossed it in the desert, right? But notice the lichen over there and some stuff over there around grows under the trash bag much better than around it. Why? Because that shields to some degree from UV radiation, and it increases the moisture underneath. Doesn't mean that we want to now trash Mars and put our plastic sandwich bags everywhere, yeah? But it shows the principle how that actually works. So I find that a cool thing. And if you're interested, we published a paper on that in Astrobiology. That's already a little while ago, but I think it's still quite applicable. Okay, let me summarize then. Habitability extends across multiple scales, from microenvironments such as water droplets to the global scale. So you have to really see differently. And if you look about microbial life, we have to consider, of course, the microbial scale. Because for a microbe, a little puddle is an ocean. Yeah? And you know what, for osmosis, a water droplet is like a lake or something like that. So the water droplets in the liquid asphalt lake are utilized as a mini ecosystem. And this is habitable in an otherwise pretty hostile environment. So, and one thing what I always say is that life and the environment is intrinsically interwoven. If you don't understand the environment, you can't understand life, and vice versa. So this is very close together. And if life exists on Mars, it might have adapted to the currently hostile environment by utilizing hygroscopic compounds. And as we found now with our PNAS paper, just fluctuating between active and inactive states. Okay? So that would be the conclusions. And if you're in something a bit more interested, you have a couple of books that I got out. And the latest actually, well, this is quite cool too if you like mega catastrophe, but it's a cosmic zoo, complex life on many worlds, where we make the point, if a planet stays habitable long enough, then it's likely to involve complex life, animal or plant-like life. That means it doesn't have to look like animals or plants, but it has the same functions, because there's many routes to it to overcome the key transitions of life. So once you have it originating somewhere, you go all the way up. The question, of course, with technologically intelligent life, that has with us only happened once. That we can't say for sure. And the last part is really discussing the Fermi paradox. But the rest, I don't think that is a problem. And we can discuss this later if you like to, or send me a note or something what you think, or if you disagree, I'd like to hear it too. Like Julie said before, I have also a science fiction novel on the left, but the latest is really the Cosmic Zoo. And I think we put there the hypothesis out that life is common and complex, and that is directly counter to the before rare Earth hypothesis, that Earth is so special. We basically say that, yes, Earth is in a way special, you know, Beethoven was special, but there were many other composers. There's many other ways how to do it. And then finally, I thank the European Research Council, actually, who generously supported that research. Otherwise, we could have never done that in the Atacama Desert. Thank you.